Alright, Assalamualaikum to all students. Okay, so I address me as Syaril. Okay, I will be your demonstration for this lab. Okay, for MIC272, it's a general molecular biology. So before we start our lab sessions, I would like to discuss with you the lab infographic reports. Okay, what you have to do for this one. So it's very important for you all to know because this one will giving you a certain portion of your courses for MIC272, right? So the instruction that you have to know, you have to form a group of 2 students, which mean kalau kalau punya group ada 30 orang, bagi pada 2 setiap group tu, right? So lebih kurang dalam berapa orang? Kalau 30 bagi 2, lebih kurang dalam 15 orang, okay? So 15 report, okay, right? So you are given permission to do by pair, right? Then number 2, the lab session topics that will covered in MIC272 is our experiment number 1, which is today, DNA extraction and estimation of DNA and the gels. Okay. So next week you will learn about plasmid extraction. Okay, and the weeks after that you will go for digestion of DNAs. Okay. Transformation of bacteria. Okay, polymerase chain reaction, or in other words, we call it PCR. The last one will be your DNA and protein sequencing. Okay. So number 3, you have to make one fully infographic posters, which include all the experiment 1 until 5. So the poster is more on the expected results. Okay. So the question at the end of your experiment will help and guide you. So please make sure awak jawab soalan semua dan masukkan ke dalam awak punya infographic poster tu. Right? So which means that experiment number 1 sampai number 5 ni, okay, you have to put inside your infographic poster. Okay, whereas experiment number 6 is just for your individual tasks. So awak kena hantar juga. How? By answering all the question in experiment number 6. So this one awak kena hantar juga. Okay. So number 4, please include your details in the assignment at the bottom right of your posters or infographic posters. So what you have to write is your name. Okay, so it means that you are in pair. So please write both name. Okay, student ID, matrix number, and a group. Okay, for example, your group 4A1. We say you 4A2, 4A2. Okay. So you are be given 2 weeks to complete this task and upload it in Google Classroom at week 15 of lectures. Okay, minggu ke-13 dah kena hantar semua ni. So please use your lecturer Google Drives. So since I'm just demo, so please make sure all these infographic poster uploaded inside your lecturer lah. Okay, Dr. Muaz, right? So please bear in mind, this task will giving you about 50%. Okay, in order you to get good grades, or even you want to score this course, you must get 15% solid. Right. Right, number 7. Okay, since we still on ODL, so it doesn't mean that you cannot attend the class. Okay, so please attend the lab session. So it is very compulsory. It means that not attend consider no marks. So I will taking your attendance via future discussion. So saya akan masukkan soalan dekat session setiap kali kita buat lab session. So boleh jawab in between our time between 2 to 5 AM. Okay, boleh jawab soalan kat situ. Okay, dan make sure awak jawab dan make it only one group dalam tu. Okay, klik jawab soalan tu dan jawab. Okay, jangan tambah soalan kat situ. Sorry, jangan tambah jawapan kat situ. Okay. Because bila saya download nanti, dia akan dapat satu senada nama yang solid lah. So make sure awak tahu cara nak guna dia. So no attend from that, it not consider no marks for your lab report. Okay. And last one will be your good luck. Okay, is there any question about your assignment so far? So ada soalan tak tentang awak punya assignment?
Tak lagi, sir.
Faham semua? Macam mana nak buat awak punya infografik tu?
Faham.
Okay. So more on expected result setiap awak buat tu. Dan juga explanation mengenai setiap lab yang awak buat. Okay. Kalau tak ada, kita boleh sambung untuk kita first session kita. Alright. Right, welcome to our experiment number 1. So make sure you have books. Okay, awak kena beli buku manual tu okay, dekat Shopee. So hopefully your lecturer dah bagi link tu Okay, kalau tak ada link awak boleh minta saya Alright. So link pada Shopee tu. Okay. So we go for the experiment number 1. It's a DNA extractions, estimation of DNA concentration and gel electrophoresis. Okay. So the first video awak tengok tadi tu adalah example of how DNA were extracted. Okay, so DNA extracted daripada they saw all a lot of cells, for example from onions. Okay, so for this experiment we're using human cells. Dari itu plant cell. Okay, so what are types of human cells? You have to check in this experiment. Okay. Okay, there will be 3 activity inside this experiment number 1. Okay, number 1 will be your DNA extraction, how you extract DNA. Okay. In other words, DNA is a process of purifying DNA from the cell using a combination of physical and chemical methods. So what's the meaning of chemical and physical? Physical is you doing it physically. Okay, macam awak korek ke ataupun awak potong ke, that was physicals. Chemicals, the process of something bahan solvent or solution. Okay. So the purpose of extracting DNA is to obtain genetic material in a relatively purified from which can be used for further investigation such as PCR or polymerase chain reaction. Okay, and sequencing. Okay, you get the sequence from the genetic genes. Okay. There are 2 major parts in extracting DNA. Number 1 will be cell lysis, and number 2, solubilizing DNA, and last one to removing all the contamination proteins. Okay, there are 3 parts in doing the DNA extraction. Okay, RNA or macromolecule by using enzymatically or chemical methods. To do the DNA extraction, you have 4 steps using to remove and purify DNA from the rest of the cell, including lysis. Okay, lyse something ataupun break down something, precipitate the material inside the substance. Okay, and then you have to purify it. Means that you have to remove all the contamination stuff like protein, RNA, and so on and so forth. Okay, and then resuspend the product you need. Itu adalah awak punya DNA, alright. So objective number 1, to isolate DNA from animal cells. So today we're using the chick cell, okay, cell PP, right, and bacterial cells. So there are 2 types of species. Okay, number 1 will be human, number 2 will be bacterial cells. Okay, so bacterial cell can be anything. Can use bacillus subtilis and so forth. So animal chicks kita gunakan kita punya cell PP, right? And then last one, and number 2, to compare the effectiveness of method for extracting the DNA. Okay. Okay, the material that we use is chick cells. Okay, so please make sure they are fresh. Okay. Number 2, fresh overnight bacterial solution. So there are the apparatus that we use is Eppendorf tubes, okay, micropipettes and micropipette tips, vortex for mixer, centrifuge, okay, and last one will be cotton swab. Okay. So chemical that we use is sterile distilled water, so make sure it's sterile, okay. saline. Okay, the difference between sterile distilled water is for washing. Okay, once you get the DNA, you use the sterile saline. Okay, denazole, direct solutions. Okay, cotton swab or plastic stirrer. Okay. Method number one, how you extract DNA from the chick cells. So first of all, you have to prepare 1,000 microliter of sterile saline. Okay. Why we need sterile saline? To make sure the chick cell is in isotonic form. Okay. Into the clean appendix tube and label with your name. So labeling is very important for all kind of works. So how? Gently scrape inside your cheeks a few times with the cotton swabs. Okay, ada setengah pakai penyukir gigi, pencongkil gigi boleh juga. Okay, tapi untuk lebih baik supaya tak melukakan awak, kita pakai cotton swabs. And then swing the cotton swab in the label tubes of the step 1. Okay, and to remove the chick cells. So make sure your solution or sterile saline jadi cloudy. Macam ni tak tahu kita punya sel banyak dalam kita punya appendix tube tu, kita make sure from clear to cloudy. So cloudy, banyaklah sel apa Okay. This one, the solution will turn into cloudy. Okay, and then you can see what? Suspension of the particles. Okay. And then you have to spin the tubes in the microcentrifuge at fully speed for 30 seconds. So full speed bergantung pada apa sentrifuge yang ada. So ia lebih kurang 5,000 G. Okay. So you have to discard the supernatant first and then wash the cell pellet with the sterile saline. And then spin again to collect the cells and decant off as much of supernatant you have. Okay, so jangan buang bahan dalam tu kita buang cecair supernatant. Okay. Resuspend the cell pellet in the remaining fluid. So make sure nombor 7 ni, bila awak dah spin 2 kali, 4 and 6, awak akan dapat pellet. What's mean of pellet? Pellet is something yang drop ball pelekat. Dia lekat pada awak punya dinding apa tu? Awak punya dinding appendix tube. Okay. So kita buang cecair yang tak bersama dengan pellet tu. So pellet tu dikekalkan. So pellet itulah kita consider sebagai kita punya DNA. Okay. So how we suspend ataupun kita kita awet atau kita jaga kita punya sample, kita gunakan denazole direct solution. Okay, so this one adalah untuk preserve our DNA. So how about we extract from bacteria? It's very simple. Okay. Number 1, take 1 ml of bacteria suspension in the broth. Okay, so you can use broth, you can use petri dish, okay, ataupun agar. Okay, so agar awak scoop dia pakai inoculum tube, okay, inoculum loop, right? And then centrifuge about 15,000 G. Okay, so this one is the same speed like this one, full speed. Okay, the same for extraction of DNA. So same speed. Okay. So, but the difference is this one we're using 3 minutes. Okay. And then discard the supernatant completely, right? So bila dah discard supernatant, which means something mesti ada yang membentuk pellets ataupun ada bahan yang termendak dekat dinding kita punya centrifuge tube tu. Okay. So suspend the cell pellet with a 500 microliter of sterile distilled water. Okay, this one we just using the sterile distilled water instead of normal saline. And vortex vigorously. Okay, so tujuannya adalah untuk kita hancurkan kita punya pellets. So this one we have to boil. So for extraction of DNA from bacteria, we have to boil. So in other words, we call it boiling water. Boiling method. So we check dalam awak punya buku dan sebagainya. So extract DNA from the bacteria, we're using what we call boiling method. Okay. So we have to boil for 10 minutes and then cool them 20 degrees Celsius for 10 minutes. Okay. So again, we have to centrifuge for 15 G with a speed of 50 G for 3 minutes again. And then you will get the color. Discard the supernatant ataupun our buang supernatant. So we assume that we get the DNA template from the bacteria. So to store or to dormant our cells, we're using the negative 20 degrees Celsius temperature. Okay. So awak dah dapatlah dua extraction DNA. Okay. One from DNA from bacteria, another one from animal cells. Okay. So and then we go for the estimation of the DNA concentration. So how you know? Is it how many of DNA you get from the experiment or activity 1? Okay, so we're using what we call under acidic condition. DNA will react with the diphenylamine to produce bluish green color complex. Okay, so what's the meaning of this one? In general, the reaction is not specific for DNA actually. It's given by the 2-deoxypentose. Okay, so why we're using extremely acidic condition? It will cause deoxypentose to be converted into highly reactive beta hydroxy levulinic aldehyde, which then react with the diphenylamine to give bluish green color complex. So to do the concentration, mostly we're using what we call spectrophotometer. Okay, so we use a special wavelength. Okay, so wavelength 595 nanometer will capture all the red color. From there, you have to calculate and do the standard graph. Okay. So for further this technique, you have to see the video. So video ada dalam buku ada, I will check it. So the main objective of the experiment to estimate the amount of DNA present in the sample by diphenylamine method. So I will be giving the YouTube ataupun video punya tu link. So I will check kat situ. Alright. So the sample we need, okay, of course the sample yang kita dapat tadi, alright. So apparatus is appendix tube, micropipettes, micropipette tips, and spectrophotometer. So there's as I said earlier, spectrophotometer, we're using a special wavelength, okay. So we're using about 595 nanometer, okay. So chemical that we use, sterile distilled water, agarose gel, and TBE buffer, okay. Okay. Stock standard solution, 50 mg of DNA will dissolve in 15 ml of saline sodium citrate buffer and concentration about 1 mg per ml. Working standard, 5 ml of stock solution. Solution was diluted to 50 ml with the distilled water and concentrated with a 100 microgram per ml. Okay. So the diphenylamine reagent, we're using about 10 gram, but we have to dissolve in the concentrated sulfuric acid, which was made up to 1 ml of glycerol acidic acid. So the solution must be prepared freshly, okay? And then you have to buffer the saline about 7.4. So how you do? 0.14 normality sodium chloride and 0.02 molar sodium citrate. Okay. So we go for the methods for diphenylamine methods. Pipette 0.5 to 2.5 ml of working standard solution into the 5 test tube level as S1 to S5, where concentration ranging from 50 to 250 microgram. Pipette 1 ml and 2 ml of DNA solution into the 2 test tube U1 and U5, so you respectively, and add distilled water, T represent the temperature, up to 3 ml for each test tube. Okay, set up another tube with a 3 ml of distilled water alone to serve as a blank. So add 4 ml of diphenylamine reagent to all tubes and keep the tubes boiling water bath at 36 degrees Celsius for 20 minutes. So let all tubes to be cooled and observe for formation of bluish color. And measure the color by using intensity at the wavelength at 595. And then we have to draw the standard graph. So what is x-axis and what is y-axis? So x-axis is our DNA concentration, and y-axis is our absorption. So absorption represent the wavelength. Calculate the amount of DNA present for the standard graph. Okay. So this one for manual. So manual detect untuk concentration kita punya DNA. Tapi kalau kita pergi kat mamal, that we call one apparatus sebagai gel doc. Gel dokumen So gel dokumen tu boleh digunakan untuk kita measure concentration DNA yang kita dapat juga. Right? So macam mana kita nak tahu kita punya DNA itu dapat ke tak? So we done the activity number 3. Okay, the third activity is our gel electrophoresis. Okay. So what is gel electrophoresis all about? It's a widely used method to separate biomolecules such as DNA, RNA and protein. So besides kita nak check DNA, dia boleh juga digunakan untuk RNA dan juga protein. Okay. So there are factors. Kenapa kita pakai electrophoresis? Number one would be very non-expensive lah, murah. Okay. Dan juga terdapat beberapa faktor yang kita perlukan dalam proses method ni. Okay. Number one will be mobility of the molecule in the gel, such as charges, size and shape, buffer condition, gel concentration and voltage. Okay. Semua ni adalah dia punya factors. Factors of what? Factors the mobility of molecule in the gels. Okay, so it depends on molecule apa yang letak dalam gel tu. Is it DNA, RNA or proteins? Okay. So we're using what we call agarose gel, which is contain microscopic pores, okay, which act as a molecular sieve ataupun penapis. Molecular sieve tu adalah penapis molekul. Okay. Smaller molecule move through the pore more easily than the larger ones. Okay, so from here you can have DNA, RNA or protein yang mana satu lebih besar. If you say you put protein akan lambatlah masuk ke dalam, bergerak. Okay. So molecules can have the same molecular weights and charge but different in shapes. Okay. So molecules with the compact shapes, for example, a sphere is more compact than a rod, can move more easily through the pores. Okay. So kalau bentuk rod atau sphere, so sphere lebih mudah lalu daripada rod. Okay. Given two molecules of the same molecular weight and shapes, okay, the one with the greater amount of charge will migrate faster. In addition, different molecules that bind more strongly to the agarose will migrate more slowly. Okay. So di sini saya bagi tahu tips adalah DNA bersifat, kena check, DNA bersifat negatif positif. Bila negatif, istilah kita letak pada kita punya bahagian positif atau negatif. Right? So the main objective of this experiment to separate DNA fragment of the different sizes on the agarose gel. Okay, awak dah dapat dari DNA awak tadi, so kita akan gunakan agarose gel untuk kita check sama ada dapat tak. And number two, to observe DNA fragment by staining with a gel star. The last one, you can also estimate the size of DNA by comparison with the known DNA ladders. Okay. So the material that we need, number one, is sample DNA adalah activity number one yang awak dapat tadi. Okay, so the difference between DNA sample and plasmid DNA, okay, plasmid DNA adalah kita gunakan plasmid DNA adalah daripada bakteria, so boleh guna juga ataupun cut of DNA samples. So mostly to compare from the experiment, kita comparekan kita punya DNA from animal tadi dan juga DNA from bacteria. Right? Apparatus, we're using micropipettes and micropipette tips. We need electrophoresis set and colloidal flask. So chemical that we use, agarose, okay, 1 times TVE buffer, gel star stains, okay, so gel star tu untuk kita warnakan kita punya samples. DNA ladder, okay, 6 times loading dye. So loading dye ni akan dicampurkan dengan kita punya sampel DNA. Okay. So gel star ni untuk apa? Gel star ni adalah untuk kita visualize our samples tu. Sebenarnya kalau kita letak 6 times loading dye ni, campur dengan DNA, kita tak nampak apa-apa kecuali kita warnakan kita punya agarose gel tu. Right? So how the process happen? Okay, number A, you have to prepare our agarose gel first. So you must make 1% of agarose gel. Okay, number 1, rinse and dry the gel casting tray with a 95% ethanol if available. Set the casting tray on the level surface. Okay. Adjust the level of comb so it rests evenly with a few millimeter of space between teeth and the tray. This will allow wells to form in the agarose. So how you do how you prepare 1%? First, you have to weigh 1 gram of agarose powder. So ada agarose powder. So awak kena mix dengan apa? 100 ml of 1 times TBE buffer. Okay. So now you have to boil in a microwave to dissolve the agar. Okay, how you know? So bila awak campurkan awak punya serbuk ke dalam cecair 1 times TBE buffer, dia akan jadi cloudy. So once you know, macam mana awak tahu awak punya agar dah masak? Cloudy to clear. Right? So cloudy to clear, maknanya awak punya agar dah pun masak. So you have to let it cool bench until 45 degrees Celsius. Okay. So once bila dah sejuk, so cara dia sangat simple je, awak nak tahu dia sejuk ke tak. So awak gunakan conical flask untuk kita masukkan kita punya agarose dan juga 1 time TBE. That's why we need a conical flask. So this conical flask akan masuk dalam microwave. So macam mana awak tahu dia dah sejuk, awak ambil conical flask tersebut, letak kat telapak tangan awak. Kalau rasa suam-suam, kita boleh mula letakkan kita punya 2 microliter of gel star solution. So 2 microliter gel star solution is very important. Okay. Kalau tak, awak takkan dapat apa-apa visual dalam awak punya agarose gel. And then pour 25 to 30 ml agarose into the casting trays. Okay, the casting trays. So allow the gel to solidify about 10 minutes and then remove the comb and tips. Okay. So why we need comb? Comb to make a well. So kita masukkan sampel dan kita campurkan DNA bersama dengan 6 time loading dye. Okay. Why you need tips? Tips untuk kita halang daripada kita punya agar daripada terkeluar daripada kita punya tray. Okay. So now you're going to load something inside the gels. In other words, loading the sample inside the well of the gel and then run the gels. Okay. So we need about 2 microliter of loading dye to each of the samples. So all loading dyes have the dense and or viscous substance. So apa yang ada dalam loading dye is about 40% of sucrose or glycerols that give your DNA sample weight so it sinks down into the well. So yang sinkkan kita punya, yang gerakkan kita punya DNA bukan DNA sendiri, bahan di dalam loading dye itu. So loading dye itu akan bergabung dengan kita punya DNA dan dia akan dibawa bergerak. Okay. So bila dia bergerak, gerakan tu awak akan tahu bahawa berapa berat ya, bergantung kepada kita punya ladder. Okay. Insert the casting tray with a gel on it into the electrophoresis chamber with a well closest to the negative black electrode. Okay, so dekat tray kita punya electrophoresis tu kita ada bahagian yang positif dan negatif. Mostly negatif berwarna hitam, positif warna merah, bergantung kepada pewarnaan. Okay. So gradually add 1 times of TBE. What is TBE? Trisborate ADTA. Okay. Or we call it electrophoresis buffer. Okay. So we have to soak our gel. Okay, kalau tak dia tak boleh bergerak. Kalau kering tak boleh bergerak, dia kena ada cair. So the chamber until the buffer just cover the top of the gel. So dekat kita punya electrophoresis tank tu, dia ada dia punya sengat ataupun penanda supaya air takkan ataupun buffer takkan melimpah. So make sure maximum. That's itu je. Alright. Carefully load onto the individual wells yang kita buat pakai comb tadi. So taking care not to puncture the well bottoms, so do not attempt to load samples. Also, if a well has a air bubble in it, push it out using a clean tips before loading a sample in it. Okay. So bila kita dah siapkan gel tersebut, kita masukkan ke dalam kita punya tank, okay. So kita load water, kita load kita punya buffer. So insyaAllah well awak tu akan dipenuhi dengan bubble. So that bubble awak kena buang menggunakan tips. Okay, so when you're loading the sample inside the well, make sure tak tertolak dia punya lubang. Well tu sebenarnya bergantung kepada kita punya size comb. So mostly adalah comb, bahagian comb yang halus di bawah tu. Close the cover and connect the electrode and run the gel around 90 volts for 60 minutes or 1 hour. Make sure the cord are correctly plugged into the power supplies, red to red and black to black. So mostly kita punya electroporesis set tu berwarna red dan hitam. Okay, red and black. Okay. So black represent negative, red represent positive charges. And then electroporesis run until the bromo, phenol blue have migrated to within 3/4 of the positive electrode end of the gel. So kalau kita ada gel lebih kurang 3/4 daripada gel lah, barulah kita boleh angkat dan kita boleh visualize. Alright. So shut off our power supply, unplug the lead and unplug the power supplies, leave the gel casting tray from the chamber. Okay, kita keluarkan kita gel dan kita bawa dia ke proses seterusnya. Okay. And then you have to stain and photographing the gels. Okay. Remove the gel from the casting tray and place it on the UV trans illuminators. Okay. So dekat lab makmal biologi ada dua bahan. Satu kita punya UV trans illuminator yang biasa punya. Yang kedua kita ada gel dock. Okay. So observe the DNA bands on the gels and take the picture with the cameras. Okay. So from that, you can estimate the size of the DNA band by comparison with the known size of the DNA layers. Okay. So from that, you have to plot the graph. Okay. So graph molecular weights of each band of the DNA layer against distance. It traveled from the well by using the centimeter. So we have to use this standard curve to estimate the size of the bands. So from here, molecular weight represent our y-axis, whereas the well distance represent our x-axis. Okay. So for the process here, you can see the video from this link. Okay. All right. Is there any question? Before we go for the lab questions, ada soalan tak? Is there any question before we go for the question in our experiment? Ada soalan tak sebelum saya masuk ke soalan lab?
Tak ada, sir.
Tak ada, alright. Ada soalan? Tak ada. Okay, so untuk further awak boleh tengok video yang ada dalam tentang proses electrophoresis dan resection, right? So saya dah bagi link kat sini, awak boleh tengok nanti. Okay, this is the question that got you, okay, for your infographic report, okay, poster report. So number 1, state the location of the DNA in the bacterial cells. Okay. Identify the main function of gel stains in the gel electrophoresis. Show the direction of DNA migration when electrical current is applied to the gel. Okay, and why they move toward these pools. Okay. 4, name any latest technology to quantify the DNA concentration. Okay. Okay, is there a question? So kita dah tamat untuk eksperimen pertama, alright. So is there a question for students? Ada soalan tak? Ada soalan?
Ada sir.
Tak ada. Okay, so untuk eksperimen pertama ni awak tahu kan cara nak buat dia punya apa tu, poster Okay, poster tu awak kena buat untuk apa, lebih kepada proses kalau aktiviti tu ada 3, so awak summarizekan tiga aktiviti tersebut dan buat expected setiap result. So jawab soalan untuk membantu awak menjawab eksperimen pertama. Boleh? Okay, ada soalan lagi sebelum kita tamatkan kita punya eksperimen pertama ni? Ada soalan tak? Any questions?
So yang soalan lab tu untuk eksperimen 6 ke ataupun semua eksperimen kena jawab soalan dia?
Semua eksperimen ada soalan. Semua eksperimen dalam buku awak beli tu ada soalan yang awak kena jawab. Daripada 1 sampai 5 except for eksperimen number 6. So tak payah masuk dalam awak punya poster. Eksperimen 1 sampai 6 ada soalan. So awak kena jawab soalan tu dan gunakan untuk awak jawab, untuk awak gunakan dalam proses untuk buat infografik poster tu. Okay?
Okay.
Alright. So infografik tu dibuat dalam satu poster Awak kena ingat boleh, one poster include all the experiment. Awak kena buat satu je poster. Tak perlu buat banyak poster satu poster je untuk course ni nak MIC272. Dia bukannya satu poster untuk satu eksperimen Only one poster include all the experiment number 1 and 5. So that's why kata tips dia adalah awak lebih kepada expectation result. So the best way awak jawab soalan. Okay. Dan juga awak cerita summarize of each of the experiment. Okay. So any question left? Ada soalan lain tak?